Good evening, everyone. Welcome you all to the ninth day of the seminar on Science, Technology and Society, the promise for next generation, under the umbrella of Azadi Ka Amrit Mahotsav. So as we all know that this Mahotsav has been inaugurated by Honorable Prime Minister in order to celebrate and commemorate the 75 years of India's independence. To celebrate this monumental occasion, the Neurocomputing Lab, Department of Electrical Engineering, IIT-Delhi, is conducting a series of talks onto the theme of science, technology, and society in order to discuss the recent technological advancements in the area of science. So today, for this evening session, we have with us Professor Srinivasa Chakravarthy. Professor Chakravarthy is currently working as a professor, Department of Biotechnology, Indian Institute of Technology, Madras. Sir's research interests include computational neuroscience, computational cardiology, and handwritten character recognition. Sir has more than 240 articles published in reputed journals and conference proceedings with more than 2,800 citations. Sir has been actively working in many projects funded by the national and the international agencies, which include the stroke rehabilitation using VR-based gaming and computational modeling, a solution for online handwritten character recognition, modeling neural oscillation, modeling vision, modeling the basal ganglia to understand Parkinson's disease, and many more. Another interesting thing which I would like to share is that Sir also works on popularizing science in Indian languages. Sir and his team have written about 75 books in Telugu, which include both the original and the translated versions in the areas of science, education, and world history. With the help of government funding, these vernacular popular science books are sent to hundreds of rural schools in the country. Indeed, it is an admirable work. With this brief introduction, I would now request Professor Chakravarthy for his talk. Thank you so much, sir, for joining us, and we can initiate your session, sir.
First of all, let me thank Professor Tapan Gandhi for giving me this opportunity to talk on this occasion of this workshop on science, technology, and society, the promise for next generation. Since I work in neuroscience, I'll talk a little bit about neuroscience. And so unlike in a technical talk, I'll talk a little bit about the societal and human aspects of neuroscience. So I'll celebrate neuroscience because I'm a neuroscientist, but I'll also point out certain serious challenges or fundamental challenges the field is facing, that in spite of all the effort and all the activity that is going on all over the world. So I think neuroscience right now is there's a lot of buzz and a lot of hype about neuroscience, and you see articles on neuroscience showing up in the media, very frequently. And probably this whole new momentum has started in the 90s when the American government has declared the 90s as the decade of the brain, and a lot of funding was invested into neuroscience research. Then subsequently, Europe also followed with a large brain project like the Human Brain Project. And the number of researchers is increasing every year. And if you go to a large conference like Society for Neuroscience, SFN, you'll be shocked to see that there are about 30,000 participants in one conference. So there are not even many venues all over the world that can host the conference of that size. And the large number of brain centers are coming up all over the world. India also we have a lot of centers are coming up. So in spite of that, if you look at some of the brain disorders, unless it is it's an infectious disease where there is a bug that is involved and there's a germ and you have to find a drug which can kill it. So then things are different. Or there is a problem which involves some kind of surgical intervention. You just open the brain and cut out the problem, cut out the tumor, for example. So then it's different, but otherwise, most complex brain diseases, right now you can say they are not curable. You can manage them, you can give palliatives and somehow let the patient learn how to survive, right, and live with it. But otherwise, there is no cure. So you see a huge dichotomy. On one side, there's huge amount of effort, number of players is increasing, and so on. On the other side, the challenges are so staggering. But I want to talk about this contrast, right? And I think we should take it more seriously and pay attention to that. And I also try to, this is the social and human side of neuroscience that we need to pay attention to. And also try to suggest that there's a need for a paradigm shift. there's need to reorient ourselves a little bit in terms of how we approach neuroscience problems. I mean, that's my opinion. I mean, whether it's right or not wrong, we will have to see. But no, that's what I'll talk about. So in the first part of the talk, I'll talk about this contrast. And second part of the talk, I'll talk about some of the work that's happening in my lab, according to as per this philosophy of brain modeling. So I said that there is a lot of hype, about neuroscience and brain in popular media. So you will see, for example, statements like this in popular media: the number of neurons in the brain are more than the number of stars in the Milky Way galaxy. So first of all, factually, it is not true because unfortunately, these kinds of statements were there for some time. Probably when this statement was first made, there were about 100 people thought there were about 100 billion neurons in the brain, also 100 billion neurons in stars in the Milky Way galaxy. But the thing is, the number of stars estimated in the Milky Way galaxy has kept going up. Now people talk about 400 billion, whereas the number of neurons in the brain is revised downwards. Now people say they're about 85 billion. So first of all, factually, it's not true. Even if it is true, if it were to be true, I would like to ask, so what, right? It's like saying the number of sand particles in Marina Beach in Chennai, right, is more than the number of stars in Milky Way. I mean, these kinds of comparisons. It looks like something big is being said, but actually, I don't know what is being implied there. Similarly, look at this another very popular statement you will see in media: the brain is the most complex object in the universe. Now, first of all, brain is a complex object, and nobody is contesting that. But how can you say that it's the most complex object in the whole universe? Because how much of the universe have you seen? How many objects? What variety of objects do you know in the whole universe? Right? We hardly even know planets in detail, right? And if you look at other star systems, so how can you talk about an object which is expected to be a small something of a small size? So this is like a 2-year-old saying, "I have the greatest dad in the whole world." I mean, a 2-year-old talks like that, because with a lot of hyperbole. I mean, a scientist who is like a rational human being, we shouldn't talk like that. But this is quite common in popular media. So I would like to ask, why is there so much hype? I mean, brain is important, neuroscience is important. I mean, nobody is doubting that. But why the hype? So there are many reasons to that. So many reasons why we approach things in a certain fashion. So first of all, we are living in the era of large projects. I mean, unless a project has a very high value, people don't even think it is of much use, right, to the society. But so that's the kind of mindset that you see these days. So therefore, when you have a large project, what happens is large projects generate a lot of data, right? And when there's a lot of data, you write about the data in popular media. And when the media talks about all this data, the project becomes popular. I mean, it looks like a lot of things are going on. And then you show all that stuff, and then go back to the funding agencies, ask for more funding. So this becomes a cycle. But this is how a large project operates, by showing a lot of output. Now, when we talk about data generation, so the thing is, data generation is becoming more and more important in neuroscience, not only neuroscience, and I think most biology. And this trend is even extending to other branches of science too. But definitely in biology, because biology is essentially by its very nature, right from the beginning, it's very descriptive in nature. And on this slide, you see a picture of microorganisms, right, hand drawn by Antoine von Leeuwenhoek, after based on his early observations with this crude microscope. So he saw certain moving, jiggling things in the slide, and he thought they are recognized that they are living organisms, but also the pictures he has drawn is is added out of imagination. And they're not, bacteria don't look like this. So he called them the animalcules. So that is animals and molecules combined, like a portmanteau word. So in the early days, people would draw pictures. These days, you use expensive equipment to make observations, and it's you can say it's a more fancy way of drawing pictures. But collecting data is very common, and describing the system that we are studying is in great detail. It's a very common tradition in biology. Now, this tradition of biology must be reviewed and see where it is leading us. In that context, I want to refer to this nice article by Yuri Lazarnik. The article is titled, "Can a Biologist Fix a Radio?" or "What I Learned while Studying Apoptosis." So he asked the question, right? And if you want to fix a radio, how does a biologist fix a radio? And this is not a joke, but this article is quite funny. So in biology, whenever you want to study something, right, you take we bring sort of a lot of copies of that. Let's say you want to study a certain gene, right? You need to, have a lot of animals in which that gene is mutated or something like that. Or you want to study certain cell, you get a lot of cells of that type. So whenever you want to study something, you take a lot of copies of that, and you study each copy. So similarly, if you want to study how a radio works, as a biologist, you would naturally order a lot of printed circuit boards of that radio, and then you begin your study. How does the study proceed? And this is what the paper says. I'm not making this up. So you look at this PCB and you say, okay, there are certain blue cylindrical objects with a silver head, right? Okay, so then I measure the diameter and note down the dimensions. Then there are other brown objects which are ellipsoidal, and it's actually a prolate ellipsoid and not an oblate ellipsoid, whatever. And then the other objects which are cylindrical, they have colored stripes on them. Then there are other cylinders which have some red wire wound on them, and I keep on describing all the different components and their sizes and measure them very meticulously and make a kind of a list of all that. So this kind of a study, in biology, would call it the structural stage, right? So this will take a whole lot of time. If you want to study different kinds of PCBs like this, it will take a lot of time. So at the end of this, you would still not have understood how a radio works. First of all, just forget about fixing it, because to understand how radio works, you have to study its function. So how do you study function? How do biologists study function? So you take this PCB, connect it to power, and then power it up, and then okay, you'll see some sounds coming out, right? Now you try to pull out components one at a time. So it is all things being equal, and that's a phrase that we use, right? So you pull out one at a time and see what is the effect of that deletion of that component on the sound produced. This is very similar to what we do in so-called knockout gene studies. Right, you knock out one gene and see what is the effect of that knocked out gene on the animal's phenotype. Right, so you do this and keep on doing this, and so let us say after some time, some smart chap finds out that if I pull out a certain important wire, probably the wire that carries power from the power lines to the PCB, the sound completely stops, and that will completely arrest the function of the radio. So that becomes a very big discovery, and that postdoc or whoever has discovered that will get a lot of job opportunities, will get faculty positions in top schools and a lot of NIH grants and things like that. I mean, this is the article makes fun a little bit about how biology research proceeds, but there is a grain of truth in it. I mean, that's actually how biology research proceeds. Now, since it's an electrical engineering department that is offering this workshop, and I'm an electrical engineer too by basic training. So when you ask the question, how does a radio work? I mean, this is the outline, the conceptual outline that you will start with, right? So first of all, we need to have some notion of an electromagnetic wave, and you should have the idea that there is something called a carrier wave and a message signal which rides over the carrier wave by some kind of a modulation technique. And that carrier is picked up by a resonant circuit called a tuner circuit, which you can tune to any desired frequency by varying some parameters. So once the signal is picked up, then you demodulate it, right? Extract the message signal, and then you amplify it and feed that amplified signal to a speaker. This is the overall outline, and rest is detail. I mean, what kind of amplifier do you want to use? What is the actual circuit relation of that amplifier? What is the tuner circuit? All that is detail, but the overall outline, broad outline is this. And any radio manufactured by any company will have to fall in this kind of a broad framework. So once you know this, rest is detail. You can master all the complexity once you have this kind of a conceptual framework. But unfortunately, when we talk about brain, we don't have any such conceptual framework. So we depend heavily on data, and we hope that the data speaks for itself and gives you magically that conceptual framework. I mean, it's supposed to be expect that miracle to happen, but it doesn't happen very often. Or rarely does it happen. So let us talk about this now, data aspect of neuroscience. So first of all, we are living in the era of big data. So in every field, we like to generate mountains of data, and then it is somebody else's problem to analyze the data and find out what the data is talking about. And data has also become one of the pillars of modern science, because traditionally, if you look at the pillars of science, a very ancient pillar of science is experiment, because Everything began by observations. Then man started asking questions and applying logic to why things are like that and so on and so forth. So that brought in the era of theory. The era of computation is much more recent. I mean, about 50 years ago, 60 years ago. And the most recent pillar is the pillar of data, right? So because and because that is the spirit of the times, because we have also very sophisticated equipment by which I can study various biological systems in the brain and at various scales and generate data. The mountains of data is being generated as we are speaking in various famous labs in the world, at molecular level and cellular level and behavior level, systems and imaging and name it. You have lots of resources and you can generate a lot of data. So this is become a common thing. So, for example, if you look at cell level, right? Your brain is a network of neurons called cells called neurons, and neurons are they come in wide variety of shapes or morphologies, and people have studied these morphologies and have even published atlases. Like, for example, by morphology I mean like a given neuron, how many wires does it have, and what is the average length of this wire, and what is the branching pattern, how does the thickness vary as you move away from the cell body, and what are the conductances and distribution of ions. I mean, name it, right? Infinite amount of detail is being explored and studied and tabulated, and people had published atlases of this kind. That's available now. Or take much larger questions of the brain, like right, the entire brain circuitry. What is it like? Can we understand that? So there is a huge, ambitious project called the Connectome Project, which aims at working out the entire wiring diagram of the brain. So let us say human brain has about 100 billion neurons, adult brain. And each neuron is connected to about 1,000 to 10,000 other neurons, right? So you're talking about 10 power 14 type number of connections. Now the objective of this project is to work out this entire graph. That is ith neuron in this huge network connected to the jth neuron. I want to work out the entire graph. It's extremely ambitious, obviously. So people said, let us wait for a little bit, in working out the connectome of the human brain. But let us begin with smaller organisms. So, earliest work was done on this small worm called the C. elegans, more popularly called a nematode. This worm has only about 302 neurons and about 7,000 connections. So nervous system of this tiny worm has completely worked out, and a lot of interesting research is happening right around this worm. So people are also trying to figure out the graph, the connectome of human brain. But the current studies do this at a slightly coarse grain level. So you don't ask whether the two neurons are connected, but you ask whether two brain regions are connected, and what is the kind of density of the fiber bundle that connects those two brain regions. So this is a very fancy project, and a lot is written about that. There is a very nice book by Sebastian Seung on this whole connectome problem and so on. Now, one would like to ask, right? Why connectome? Right? Why do we even want to understand the brain's connectivity in great detail? Because first of all, I would like to know as a scientist, conceptually, the underlying reason behind the overall architecture of the brain. For example, why does brain have two hemispheres? Why two? Why not four compartments? Like the heart, or six compartments, seven? Why two? What's the logic behind it? So another major feature of brain's anatomy is this sheet of cells called the cortex, which surrounds the entire brain. Why is it like that? Right? Because if I just want, if you ask, give me a chance, I ask me to design a brain, I'll just take a bunch of neurons, connect them in some random fashion, and just shove it inside the skull. Why have this sheet of cells surrounding the entire brain surface? Similarly, if you look at some brain structures, a lot of brain structures are inside this brain volume. Some brain structures are actually outside, as if they're some kind of an appendage, like cerebellum, for example. Why is it outside? Why is thalamus and the brain structure inside? So I don't even understand the basic underlying logic of brain's organization. Because first I want to understand that, and then I'll get to detail. I want to understand the underlying computational principles of brain's anatomy, and I want to understand why. Then I can work out the details of the connectome. But we don't have that kind of understanding now. So therefore, we have big data. We have lots of data, and data is growing every day. But the theory is somewhat tiny. It's somewhat weak. So to understand this situation, also to get an insight into what is the way forward and how do you solve certain fundamental challenges of neuroscience, I'll like to draw analogies between the history of neuroscience and history of astronomy. Like basically, astronomy as a branch of physics. Or you can say history of physics, but I'll take astronomy as a special case. And if you draw parallels, right, that might give a clue to what kind of breakthroughs must happen, right, and where in neuroscience for us to be able to solve some of the fundamental challenges. So if you look at astronomy, right, for a long time, so people had some knowledge of the heavens, so the planetary motion and star motion. I mean, as we see it from the Earth, for thousands of years, and all over the world, ancient cultures had a reasonable amount of observational astronomical knowledge. So even about 2,000 years ago, if you look at Claudius Ptolemy, right, he has mapped a lot of stars, and we knew where they are at a given time. Only thing is, he was depending upon a kind of a geocentric model, right, and everything was going around the Earth, and the trajectories of various heavenly bodies look very complicated when you saw them from the Earth. So he thought that planets go around the Earth, but so that is, and he assumed it's a circular orbit, which is not true. And within around that circular orbit, you have to superimpose another secondary circle, right? So the planets go around this follow this kind of double cycle, very strange orbit, and then that's how they're moving. So that's what he has proposed. So even 1500 years, for 1500 years, people have lived with this kind of strange descriptions of planetary motion. And around, I don't know exact year, but around 1500s, right, when people like Tycho Brahe, right, got into the field, he found that the predictions of eclipses, which is very important in those days because of the connection to astrology. People used to predict the fate of people and nations based on just like it's popular even now, right? So he said it's quite unacceptable that the predictions of eclipses are off, right, by about a couple of days. So which is considered a huge error. So he said we need to fix this. And what is his approach to fixing this error? Not by fixing the geocentric model and all that. He didn't talk about any paradigm shift. He just said we need to observe more frequently. So we need bigger equipment and more resources and make more observation. Basically, big data. He wanted to generate more data, and he thought that generating more data will just fix the problem magically. So he did that. He had a lot of resources and equipment, and he made more observations and more frequently had more data. But with all that, what has he achieved? He has proposed this what is now called a triconic system, which was like a midway between geocentric and heliocentric world models, because the religion was strongly geocentric, right, in its view, whereas the data seemed to suggest more heliocentric view. So now this guy cannot pick up fight with religion and church. So he came up with a political, compromise, and came up with this kind of a midway view. So what happened to all that equipment and data, right? What has it led to? You can see it here. But on the other hand, Kepler, who lived at about the same time, and Kepler was quite inspired about astronomy even from his childhood. He as a child, he witnessed the great comet, right, of 1577. And all his life, he lived a life of poverty. He was literally hand to mouth, trying to get some sponsors for his research and for his livelihood. And he was even trying to get some data from famous and well endowed Tycho Brahe, but Tycho Brahe didn't reveal his data until the end, until he didn't share his data. So Kepler took a more theoretical approach to the problem. He wanted to understand the logic of the planetary orbits and the radii. How big are they? Because people knew about the first 5 planets, or the visible planets, for a long time, and he wanted to understand what is the size of these orbits. Can we come up with a formula for predicting the sizes of these orbits? So he was under this classical Greek influence, and which somehow thought that world is made up of numbers, and the Pythagorean school believed that right numbers govern the world and nature and the structure of the universe. So he thought that the Platonic solids, this perfect polyhedra, right, have something to do with the planetary orbits, and try to come up with a model, and that didn't work. He struggled with it for a very long time because of this early brainwashing Finally, at one point, he kind of moved away from this belief, a very long-standing belief that planetary orbits have to be circles. Why circles? Because circle is a perfect shape, and God likes perfect shapes. Therefore, heavenly objects are all perfect and divine and kind of blessed by God, and therefore they have to be. So this is a kind of nonsense that was quite popular, and Kepler was also under influence of that for a long time. But once he deviated from circle and sort of opened the possible, became open to the possibility that it could be ellipse and not a circle. Then suddenly he could find a way of accommodating a lot of observational data, and he came up with these three laws which we all studied in our high school, right? So with Kepler's, so if you look at the science of astronomy, it began with big data. It began with small data, then went to big data, and then that led to deep discovery of deep patterns in the data, and that gave rise to development of really profound and universal theory, really solid theory. And that was, as you know, was developed by Isaac Newton as a young Newton. He literally sat in his village in Woolstop and worked out mechanics and optics and a whole lot of things. We know that story, right? So what is beautiful about Newtonian mechanics is the deep generalizations, right? You see connections between apparently totally disparate things. The falling apple and a falling moon follow the same law, exactly the same law. How come? Because there's this very nice illustration given by Newton, right? To understand how moon's orbit as an object that is basically trying to fall towards Earth. So he imagines that if you stood on a mountain and threw a stone, right? If you threw the stone at not a very high velocity, it'll just fall down and fall maybe at the foot of the mountain. If you threw the stone at increasing velocities, it will try to fall further and further away from the mountain. But since Earth is a ball and not a flat surface, as this stone keeps trying to fall towards the Earth, and as you keep trying to chuck it at higher and higher velocities, you will reach a point where the stone will not fall to the Earth anymore. It will try to fall, it will follow the curve of the Earth, it will try to fall, but will never fall. It will keep going round and round the Earth. It's a very beautiful intuition. It gives a good sign of how his intuition, how powerful his intuition is, and how it works. So like that, he was able to see a deep analogy between something like a moon orbiting around the Earth and a falling apple. Okay, beyond these poetical connections, he was also able to develop the right mathematics, the calculus and differential equations, and also hypothesize something called a law of gravitation. And with that, he was able to explain the Kepler's laws, and that gave rise to what we now call the universal law of gravitation. Because Kepler's laws is still based on planetary observations of our solar system. But Newton's law is called a universal law of gravitation because look at the confidence of that man. He believed that this should be happening anywhere in the universe, right? I mean, that's science, that's fundamental science. But we haven't reached that stage in neuroscience. We don't have any such one. We have some, but there's some you can call fundamental laws. We don't really have. Somebody asks you what are the 10 laws of the brain. Nobody knows. We have no idea, and that's the problem. I think that's the problem. So the Newton evolution neuroscience, we haven't seen it yet. Because on the left, you see the picture of Newton's Principia Mathematica. On the right, you see the textbook of, the famous textbook of neuroscience called The Principles of Neural Science. So it kept, so there is this British researcher called Daniel Walford who works on motor function. He was joking in one of the conferences that, so the candle and we call it candle and Schwartz. That this textbook keeps getting fatter and fatter with every edition. Thing is, if it is a book about principles, why will it get fatter? Because principles will be very economical, very small in number. So the description also should be very compact, right? If you are publishing books which are fatter and fatter in 1,000 pages, 1,200 pages, and so on, that cannot be about fundamental principles, right? So he just jokes about it. Basically, people know that there is a problem. Only thing is not when people talk about it openly. As for this, the theoreticians are particularly conscious of this problem. So, for example, and many are voicing that opinion. For example, the Indian origin scientist at UCSD, V.S. Ramachandran, he says that we are at the Faraday stage in neuroscience. What it means at the time of Faraday, we haven't yet seen the Maxwell equations. Right? Only when you have Maxwell equations, you have like a comprehensive description of all electromagnetic phenomena, and there's nothing else outside that. And everything falls within the purview of those beautiful equations. We haven't reached that stage in neuroscience. So similarly, Patricia Schosland, who is a neuroscientist and neurophilosopher, she also says that there are no neural theories or no good ones anyway. So right now what happens is, although there is so much data and so much theoretical work is also happening, we don't have any satisfactory comprehensive theory of most important brain structures. We have theories, but for every brain structure, we'll have half a dozen theories. So which one should we believe? We don't know. It's anybody's guess. So this problem is also compounded by another fundamental tenet or a principle in biology, or a philosophy that is followed in biology, which is reductionism. So biology is predominantly driven by the biochemical paradigm or biochemical molecular biological paradigm. So where we try to explain all phenomena in terms of the smallest constituent. So this is the whole philosophy of reductionism. So in case of brain, we would like to explain everything in terms of molecules and cells, which is neurons. So thing is, this kind of insistence on having to explain everything in terms of smallest constituents doesn't exist in engineering. So for example, just imagine you are an aeronautical engineer and you want to design aircraft wing. You want to study the effect of the flowing wind on the wing. What is the drag and what is the pressures and all that stuff. So imagine if like a biologist, you take a reductionist view and you say, I want to understand the momentum imparted by various air molecules on the wing, right? And you do a molecular dynamics simulation of the wind flow on the aircraft wing. I mean, you need the biggest computer possible, and it will take maybe 10 decades and decades to do a simulation like that. What is the point? Why do you want to do that, right? Because in engineering, we don't do that. We describe the wind flow in terms of something called a fluid or a fluid flow, and we use Navier-Stokes equation. Now, fluid is not molecules. Fluid is an abstract thing, actually, right? It's an ensemble of molecules, but we don't mention molecules at all in the description. But it's a rigorous description, is mathematical and rigorous. And with this approach, you can design the wing and your aircraft will fly in a finite time. So because of all these problems, the description of the theories or conceptual framework that describes the function of various brain structures isn't quite solid, right? So although you have a lot of data at smaller scales, like molecular and cell level and all that, at larger scales, in terms of our understanding of various brain structures, things are very unsatisfying. And because of this problem, even our understanding of various brain disorders, what are called multifactorial diseases which have many factors, they are not purely genetic, right? They're not because of some problem with some protein or right some cell type. It's not just that, and they're very complex because of many interacting factors. These are multifactorial diseases like bipolar and schizophrenia, or, right? Depression and Parkinson's and Alzheimer's and so on. Right, we don't have we don't have a good theory, and therefore we don't even have good treatments. Right, they help the palliatives, they improve the quality of life a little bit, but it's not like malaria or something where you can give a drug and you're cured. It's not. We haven't yet reached the stage. And ironically, even in cases where certain medications work, right, they work in very mysterious ways. It's almost like magic. Take, for example, the idea of vagal stimulation. Right, there's this nerve called vagus nerve, which is a part of the parasympathetic system in the nervous system, and this nerve goes to many parts of the body, including the heart. And if you stimulate the nerve, heart slows down. Now, somehow people figured out that if you stimulate this nerve electrically, it will help suppress epileptic activity, or it will also help depression, major depressive disorder. Right, but why? What's the connection between epilepsy and this nerve, or between depression and this nerve? We have no idea. Right, and on the other hand, since this nerve also innervates the heart and it slows down the heart, if a kind of a parallel current signal goes to the heart because of the stimulation, it can give rise to cardiac arrest, and that's obviously quite dangerous, right? Now, how does it work? Like I said, the Wikipedia article citing this paper, Edwards et al. 2017, says as of 2017, little was understood about how exactly vagal nerve stimulation modulates mood and seizure control. So we don't know. It's just a elaborate way of saying we don't know what the hell is going on. Another very popular treatment for what is called bipolar disorder. It's a psychiatric condition. So they give this lithium, which is an element. It's not even some kind of a complicated biomolecule. And this has been given for last 70 years. It's one of the standard treatments, standard medications for bipolar disorder. So in bipolar disorder, the subject shows extreme mood shifts, right? For example, mania is like, the person is very exuberant and he thinks he's on the ninth cloud and all that. That's called mania. And after some time, the person goes to the opposite extreme of depression. So they keep cycling between these two extreme polar emotional conditions over a time period of weeks and months. So this drug is supposed to help that, but you see that last statement. Exactly how lithium works to do this is not known, though. Right? As I'm quoting from this website. So it is not just a few stray examples. I mean, this is a book on psychiatric medications or psychiatric treatments. It's called Prescriptions for the Mind. It's written by a noted psychiatrist called Joel Paris. In that book, at one point in a chapter on psychiatric medications, he says he was talking about his own his own training as a psychiatrist, and when he just began his practice around 1972, he's talking about situation at that time. Psychiatrists had a useful armamentarium of drugs falling into 4 groups: antipsychotics for schizophrenia, tricyclic antidepressants for depression, lithium for mania, and benzodiazepines for anxiety and insomnia. So, which is great. With all these agents, we could help many, if not most, of our sickest patients. So he's celebrating, right? We have such wonderful drugs, 1 to 4 important classes of drugs, which can be applied to a wide variety of conditions in psychiatry. But in the same page, a little below, he says this. One should also keep in mind that we do not know how most of the drugs used for mental disorders work. Most of the drugs is not just 1 or 2. It is entirely possible, however, that advances in neurosciences may lead to further breakthroughs in pharmacotherapy. Just a second. So I tried to show that on one hand, we don't have satisfactory treatments for most brain disorders. On the other hand, wherever we had good medications that are available, we don't know why they work. Right? We don't have the mechanistic understanding of their action. Now, I'll just turn up from I'm not a doctor, not a clinician. My basic degree is engineering. But whatever I've seen in my personal experience of those I've met or those family or friends and all that, it is quite staggering. I mean, what brain disorder can do. I'll just narrate a couple of examples because I could have given statistics, but statistics are too cold. I mean, I don't have any emotional reaction when I see statistics in epidemiology. So I'm not citing in epidemiology. So once I was traveling on a ferry, in San Francisco, traveling to see one of the bridges. I saw this woman, a middle-aged woman, with some book on neuroscience in her hand. I thought it's interesting. It's not a textbook, it's a more general book on neuroscience. So I just started chatting with her and she was telling her story. So her husband had a stroke 18 years ago. And since then he was kind of confined to a wheelchair. So this lady has to, help him, with his food and, with his bathing and washing and all that. And has to take him around in a wheelchair. And she's a middle-class woman. Okay, she's not doesn't come from a very rich family. And she has two daughters, and she had to go to job and make money. And she struggled a lot and was able to bring up those 2 daughters. Now they have settled and become independent. But I mean, when he was telling her story, I could see the amount of struggle this woman had to go through, because there's nothing you could do, right? I mean, at least for the 18 years was like that. 18 years of trauma, and there's no way of just fixing that person, making him normal. So and another serious challenge is mental retardation. So in olden days in India, right, people used to have a lot of children, and each sometimes we have like 10 siblings in the same family. So often in the such conditions, the last sibling, right, would have been born, with some nutritional problems and all that. And mental retardation is quite common. So I know one such person in the family, and he did not go to school beyond about, I think, fourth grade or so. He's now like in his mid 70s, and all his life he was dependent. And for the last 10 years also, he even needs a helper for his bathing and feeding and everything. He just doesn't want to eat, doesn't want to take care of himself, right? So he hasn't learned anything. He cannot read even single letters, right? So, so a person who lives like that and is so dependent, right? I mean, what does neuroscience give in that condition? Nothing. Okay, so let me give another example again, which I've seen personally. So road accidents are very common. When some people die in the accident, some people survive, but sometimes you feel, is survival of a road accident, is it a punishment to that person? So I know one person, in the family. He had a road accident. It was a grievous accident, but somehow survived after a very intense surgical intervention. He was in coma for about 8 days, and after that, he was given very rigorous cognitive and physical therapy. In fact, throughout the first month, he could not even recognize his family members in the first week or so. But he was a doctor by profession, so he was able to recall some of his medications and things like that. But he was not able to recognize his family members, and he would behave like a small child in the way his manner of talking and all that. So it looked as though this entire trajectory of evolution from childhood to adulthood, rise, is kind of recapitulated in that 1 month of recovery. So also he's recovered and is back at work and all that, but he's no more the same person. There are serious personality problems and a lot of things he doesn't understand certain things. He's very stubborn and indecisive and so on and so forth. Right? Typical prefrontal problems. Now, what can neuroscience do? Not a whole lot. I mean, whatever is done is already a big miracle. But beyond that, there's nothing like a permanent cure for that person. So when I see all these things, right? So what is the way forward? I mean, do we just keep on generating more data and write more genetic profiling and transcriptomics and proteomics and all that, or do we look in a different direction? From my perspective, my opinion on this, because as a scientist, one has to have an opinion. One might be wrong, but one has to have an opinion, I think. Right? My view would be to look for breakthroughs in the fundamental conceptual frameworks, right? The mathematical frameworks with which we study brain. Okay, so that completes the first part of my talk. So I'm arguing for this. That is, there's a need to develop simplified models embodying the right kind of abstraction, the way it is done in physics and engineering. If this can be done on a large scale and in the right way, because there's something called a right theory. You can come up with. If you look at the astronomy again, right? People had used epicycles and they tried to fit trigonometric time series to data and all that. They are quite accurate, but that's not science. They're just curve fitting. Science is a lot deeper than that. It gives an ontology, a conceptual framework. And with the same set of concepts and equations, you'll be able to explain a wide variety of things. That is science, right? So by that standard, we haven't yet reached that kind of an in-depth understanding of brain function for most systems. So I think that is where I think one has to head. So I'll just take a 1 minute break just to catch my breath, and I'll continue. I'll talk a little bit about the work we are doing in my lab with this philosophy. I'll come back in a minute. Okay, so my lab is called Laboratory for Computational Neuroscience. So our goal is to build a simplified model of the whole brain, right? That is, so this is like a cartoon picture of the brain that we use as a reference, right? So brain takes sensory inputs from the world through your eyes and ears and all that, and produce motor output through your hands and legs and speech and eye movements and all that. So if you can think of the whole brain as some kind of a complicated deep network, if you like, because that's a very simple analogy. So these input channels and go inwards and kind of end in a big decision-making module called the prefrontal cortex, right? And then the output of prefrontal cortex goes to various other regions, and finally it percolates downwards and have influence on your motor apparatus, like your legs and hands and eyes and all those things. Now that's a big, you can think of it as a big feed-forward network. Then there are a few major loops, and on these loops you have very important systems. There are three important systems: the cerebellum, basal ganglia, and hippocampus. So if you can have a brain model which just captures this, right? It doesn't have to be very big. It doesn't have to have 100 billion neurons. My belief is if it has something like 10 million neurons, right, in a simulation, I mean that should give a lot of insight into the real brain. So we would like to construct this and use this to develop disease models and try to get more insight into disease and use that kind of computation model to create a kind of quantitative framework for treatment of neurological diseases. So with that objective, my lab is divided into many teams. Each team works in one area. So the idea is that the outputs of various teams can be subsequently taken up and integrated in order to construct this large-scale brain model. So we're working on several topics like basal ganglia, Parkinson's, spatial navigation, hippocampus, neural oscillations, neurovascular, right? Some problems in vision and stroke and rehabilitation, things like that. So we have our work on basal ganglia was published in this research monograph. I'll come back to this in a minute. And also I like to simplify brain science because, like I was saying in the beginning, there's a lot of hype and there's a lot of when you read popular media, it looks as if the brain is so complicated that you can never understand it. You're only supposed to admire it or even worship it, the way you worship a god on your altar, right? But whereas I feel you should not worship it, you just have to understand it. And if you look at the brain from the point of view of details, it does indeed look very complex. But if you look at it from the point of view of principles, it's actually quite simple. I mean, it's not that complicated. You can reduce it to certain fundamental principles. At least whatever principles we know are already quite helpful, and they really give a reasonable logical picture of the brain. There's some more that we don't know. So with that perspective, I've written this book called Demystifying the Brain, which can be accessible to both a biologist and an engineer. For a biologist, it tries to present the computational view of the brain without using math. And for an engineer, it presents neuroscience without using too much of biological jargon. Because an engineer reads a biology textbook, they are put off by all the jargon, right? Whereas in this book tries to use minimum jargon so that only the principles can be explained. So I'll talk about some of these topics. In fact, I'll skip some of these things because the actual work is not very important for this workshop. I would only like to give a perspective of some of the problems of neuroscience, especially when you see that from a societal and a human point of view. So I'll talk about a little bit about our work on basal ganglia. So the basal ganglia is a circuit. It's a deep brain circuit located, as you can see in the slide on the left side. It's set deep inside the brain. It consists of many different modules, if you like, and these modules are shown on the right side. They have different names, but it looks like a very complicated circuit there. But I can redraw this in a simpler fashion, right, as a block diagram. So in this block diagram, you see that so brain can be divided in a very simply as cortex and subcortex. The cortex is the surface of the brain, right, which covers the entire brain surface, and then all the stuff that is deep inside the brain are called subcortical structures or circuits. So basically, is a subcortical circuit, and it has many different modules in it. And so the cortex sends inputs to basal ganglia, which you can see at the top, and all these modules are parts of basal ganglia. So I'm not going into the names of the modules, and you can call them ABCD if you like. But one of these modules, which is circled here by a red circle, called SNC or substantia nigra pars compacta, This is a very crucial module. This is where cells start dying in this disease called Parkinson's disease. And because of that, you will see symptoms appearing in many domains of brain function. Because of any damage to basal ganglia, the basal ganglia has role in all the four major domains of brain function, like for example, sensory motor function, or cognitive function, or affective function, that is emotional activity, or autonomic function, that is the function of the activity of the internal organs. All these things are controlled by basal ganglia. It has a contribution there. So therefore, any damage there can affect, can be manifest in all these different domains. In motor function alone, you can, because of this disease, which affects cells in SNC, you can see impairment in how we reach out to objects by extending our hands, how we write, how we make eye movements, how we speak, and our posture. All these things can be affected because of loss of cells in this very tiny structure called substantia nigra, or SNC. And on the right bottom, you see the handwriting samples of a normal person and a Parkinson's person, a patient. You see, it's a Tamil word called Sri Rama Jayam. When normal person writes it, the letters are legible and large in size. But when a PD patient writes, they're all small and illegible. Now, this kind of a writing is called Microgravia, which is a characteristic feature of Parkinson's. So we began looking into this problem, and we wanted to understand, like an engineer would, how does basal ganglia work, and what happens when these cells in SNC die, and why does loss of these cells give rise to all these motor dysfunctions that you see in Parkinson's. Okay, so I cannot explain this in great detail, but I just like to say that we have made a hypothesis about the function of basal ganglia, which is a slight deviation from the mainstream thinking on this subject, at least at that time, And based on that, we were able to develop a unified model of basal ganglia, and with this model, we were able to explain a wide variety of functions related to basal ganglia. So we have about 30-35 journal papers on that, and so over all these papers and over a series of PhDs, these are some of the students who did their projects, thesis on this area. So we're able to explain why there is a change in handwriting, why there is a change in reaching movements, or a change in gait, in case of in PD patients. So using the same model, we were able to explain a wide variety of phenomena, And all our work is consolidated in this research monograph, which was published a couple of years ago. Subsequently, we wanted to study that. Until that point, we just assumed that these cells die. In fact, we don't know why these cells die. In fact, the literature also calls the disease, Parkinson's disease, is called idiopathic. It's just a fancy word to say I don't know. Basically, that says that we don't know why these cells are dying, so we don't know why this disease occurs. So Vignan, in his in his PhD thesis, he has taken up this study of why are these cells dying, and we found in literature that a couple of groups, in international groups, have suggested that the reason behind cell death, in Parkinson's is energy deficiency. That is, inadequate energy is reaching these cells, and these cells have very high metabolic demands because of their anatomical constraints, and they have very rich wiring, the rich harbors, which project to large number of targets, widely distributed targets, both in the cortex and subcortex. Therefore, they need a lot of energy. So they can easily be burnt out and experience a burnout because of their normal mode of operation. So Vignan, in his thesis, has done a series of simulations to show that, if you constrain the amount of energy going to these cells, in terms of glucose and oxygen in the simulation, you could see a lot of pathological changes that you normally see in PD conditions that can be seen in the simulation. So a lot of pathology that you see in the Parkinsonian basal ganglia, we were able to observe in the simulation simply by one underlying cause, due to metabolic deficiency. So the underlying cause, there can be many manifestations, but the root cause is energy deficiency. So in fact, after that, we developed this theory further and proposed that the root cause of not only Parkinson's, but other neurodegenerative disorders like Alzheimer's and ALS and others. The root cause is the same, energy deficiency. And we have written a review paper on with that kind of a perspective. So with this kind of a model, we want to put the whole problem of treatment of Parkinson's on a more quantitative footing, right? So right now, the way you assess a patient's condition is using what is called a UPDRS score, where the doctor looks at the patient and makes some observations and tries to give some kind of a score of various domains of activity of the patient. But what we are proposing is that you need to actually record data, quantitative data from the patient in terms of the patient's speech or patient's eye movements, patient's hand movements, gait movements, etc., etc. And importantly, all this data can be recorded using just a smartphone. You don't need very high-tech equipment for that. And once you have the data, we want to put it into our computation model, which we already developed. And then you invert the data. That means the data becomes, the data will be used to calibrate the model and set certain high-level meta-parameters of the model. Once you have that, the model becomes a patient-specific model. So that model replicates a patient's pathological condition. And once you have that model, whatever treatment you want to give to the patient in terms of how much drug and what drugs, and right, sometimes we could also use something called deep brain stimulation, right, where you insert electrode into the basal ganglia and electrically activate the system. So all these different tools exist, but what combination with what parameters do you apply these in this treatment protocols? That can be worked out and optimized computationally by using the model. And whatever combination works best in that model, which is patient specific, must then be given to the real patient. Otherwise, what happens right now is all these treatments are given to the patient in a trial and error fashion, because the doctor just tries drug after drug and whatever works is given. But if this whole pipeline can be perfected, then all those therapies can be first optimized and designed from the simulation, and then you can give it to the patient. So similarly, we also did a lot of work on hippocampus and spatial cells. Hippocampus is the part of the brain which is involved in memory, right? And hippocampus was popularized in India by this movie called Ghazni. I'm sure a lot of you have seen this movie. But more recently, hippocampus was made popular because of the Nobel Prize, which was awarded to work on hippocampus. So particularly this prize was given to the spatial navigation functions of hippocampus or research in that area. So in 1971, John O'Keefe and his colleagues have found certain cells in hippocampus which fire whenever the animal is at a certain location, right, in the surrounding space. Therefore, they are called place cells. And more recently, in 2005, this Norwegian couple, May-Britt Moser and Edward Moser, have found another kind of cells which fire not just at one location but at multiple locations. And all these locations form some kind of a nice periodic grid, which is often a hexagonal grid, which was quite intriguing because the brain is a very messy area, right? Where do you get that kind of a nice regularity in the brain? What is the origin of that? Which is quite intriguing. And so these two studies together were awarded Nobel Prize in 2014. So this video just gives an idea of what the place cell is. So in this video, there's an electrode stuck inside the rat's hippocampus. And the experimenter is throwing some food pellets just to make the rat run around instead of sitting in a corner. So you see that whenever the rat goes to that corner of the box, right, that red dot indicates the firing of a neuron, that particular neuron. So the neuron is firing only when the rat goes to that part of the box, and it happens to be a place cell. So because that neuron is somehow encoding that location right in the box for the animal. So therefore, because of the many such cells, because of presence of these kinds of cells, hippocampus is called the GPS of the brain. So it's like this is what tells the animal's brain, where it is located, which way it is going, and how it should find its way around in space. So since it's not a technical talk, I'll skip some of these things. We developed a model which can explain, why there are these grid cells and why we have hexagonal periodicity. It has, we have linked it to certain properties of right? We borrowed some ideas from matrix theory. So there's a class of matrices called succulent matrices. This regularity of these grid cells is coming because of these properties of succulent matrices, right? If you want more details, I can share the papers with you. And we also extended this work to 3D navigation. We got the data from Michael Yartsev at UC Berkeley, who studies navigation in bats. Bats also are mammals, but they can fly. And so they also have hippocampus. And Yartsev's lab recorded neural activity from hippocampus of bats. And bats also have place cells, that is, the cells which fire at a location. Only thing is, this location is a point in 3D space and not in 2D. So the place cells here, plus fields here are volumetric. They are 3D volumes. So in our model, which we extended to 3D navigation, there also we found place cells. But the model also found two new kinds of cells, which were not known in literature until then. So we found a kind of cell which fires whenever the agent or this flying animal, right, the simulated flying animal, crosses a certain plane, right, embedded in the 3D space. So we called it the plane cell. And then we also found another kind of cell which fires whenever the agent crosses not just one plane but multiple planes. So we call it a stack cell. So plane cell was subsequently discovered by my student Karthik, who went on to do postdoc with Yartsev in Berkeley. So when you dig up the old data from Yartsev's lab, we found actually that there are cells which can be described as plane cells. But to my knowledge, there's no evidence of anything like a stack cell. So this is one example of how theory can predict very novel things which are not known in experiment. So this work was published in Nature Communications. Similarly, we have done several other studies. I'll skip some of this data. And we're also doing some work on 3D navigation, where we are getting some inputs from Ayan Mukhopadhyay, who is a theoretical physicist at our physics department. So we are using a topic called spherical harmonics to work out this theory. It's taking some time, but we are trying to develop a very comprehensive theory of 3D navigation and hippocampus. Next, we are also doing some work on neural oscillations. So the brain is full of oscillations, right? But if you look at brain theory, and if you ask the question of what does a neuron's activity represent, right? Then people mainly talk about two different theories. One is called the rate code, other is called the spike code. But because neurons, the activity of neurons is in the form of sharp voltage spikes. You can think of them as a kind of train of delta functions, mathematically speaking. Since you're engineers, I think that analogy clicks better with you. So now, what is the exact information present in the neurons activity? So the rate code people say that it's in the spike frequency, right? Whereas the spike code people say that no, it's the information is also present in the spike to spike interval, what's called interspike interval. So spike timing also carries information. It's not just spike frequency. So there are two camps which describe the neural code in two different ways. But there is a third camp which is somewhat a smaller group, but which is gaining importance, where they believe that the fundamental unit of computation in the brain is not a single neuron, but a bunch of neurons, what is called a neural ensemble. Now, if you take a neural ensemble and you take the collective activity of all these neurons and also low pass filter it, you get a fairly smooth signal, kind of like what you see in the slide in the bottom. Right, so the signal is now very different. Doesn't look like those sharp spikes of spiking neuron. So therefore, if you want to model not a single neuron but a neural ensemble, how about using oscillators to model that? Because it's a smooth signal, it looks like an oscillation. So why not use mathematical oscillators to model that? So a lot of people are doing that, and we began with this in the same approach, but we took off in a different direction about 4 or 5 years ago. We started using something called a Hopf oscillator, which can be expressed in complex domain in terms of complex numbers. I think I'll skip some of these things, and we had some interesting breakthroughs with which we are able to generate large networks, able to train large networks and model very complicated brain signals like EEG, multi-channel EEG signals, like even up to 50 channels We could model it using network of oscillators, and we could train it to capture these kinds of signals on a large scale. We're also using these networks to model locomotion rhythms. So especially if you look at quadruped animals with 4 legs, their locomotion rhythms are quite complex and quite interesting to study. And there are rhythmic examples of nice rhythmic behaviors. So we were able to model them using these oscillator networks. And I have a student who is trying to use these models to train like a legged robot. And this work is still kind of a work in progress. We also designed something called a deep oscillatory neural network, which is like, you all know, I think, deep networks. This is also a deep network, but a network of nonlinear oscillators. And all these oscillators are complex variable oscillators. The weights are all complex, and we're able to train them, and they're very powerful at mapping input signals onto output signals. Then we also done some work on visual system. I'll skip all this. Then finally, I'll come to modeling neurovascular coupling. So in the brain, neurons to perform their activity, they need energy. Energy comes from blood vessels. Like any cell in the body gets its energy from blood vessels, because blood vessels carry glucose and oxygen, right, to cells, and neurons are no different. So we began asking this question: how exactly do neurons and blood vessels interact? Right. So way back in 2010, we started looking at this problem like an engineer would. So we said, okay, I have a bunch of neurons here, right, and they get inputs from other neurons in other parts of the brain. Now the level of input a neuron receives determines its level of activity. A neuron gets a strong input, it has high activity, and the activity level of a neuron determines the energy demand of the neuron. If a neuron is firing away at high firing rates, it becomes more hungry, it needs more energy, right? So we said if the energy comes from the blood vessels, it incidentally comes from an intermediary stage called the glial cells. Okay, so whatever it is, it comes via a network of cells. Now we propose this principle called energy matching principle. That is, the activity levels of a neuron determines its demand, must be matched by the total energy supply that a neuron receives. It's a very simple, commonsensical principle. I mean, any engineer would agree to a hypothesis like this. But that has simple hypothesis, when you translate it into other, work out the biological consequence of that, it has a lot of interesting consequences, and which completely flies against the mainstream thinking of how neurons and blood vessels interact. And it was a very difficult fight. We're still, in the middle of this fight. So I'll skip some of this stuff. And most recently, last year, we published a paper on what we call artificial neurovascular networks. I mean, as engineers, I'm sure you all know about artificial neural networks, but this is an artificial neurovascular network where there is a vascular network, basically a circulatory system, which supplies energy to the neurons. Right, that is only when a neuron receives energy, it will be able to get activated. Otherwise, it won't get activated. So now the problem is, different neurons have different levels of activation. Now, how do you determine which neuron needs to get how much energy? Because I have a total cap on how much energy I can give. So this paper was published in scientific reports, Nature Scientific Reports last year. And there's a lot of very interesting biological hypotheses coming out of it. And if you're interested, you can take a look at this. Okay, I think that's a quick overview of some of the things we are doing in the lab. So what is the road ahead? What do we plan to do in the future? So basically, with all these modeling efforts, where we looked at diverse aspects of brain function, we feel it is possible to develop kind of unified models of brain function. And there's a way of going about it. And we have reduced it to some kind of a simplified formula, which says that there are 3 stages of this development. The first stage is simplify, the second stage is unify, and third stage is apply. So how does it work? In the simplified stage, you have to have a model which is made sufficiently simple that if you make it any simpler, it looks absurd, right? So for example, if you see this picture of Mahatma Gandhi, on the left side, you have a very detailed sketch with shading and all that. On the right side, it's a simple cartoon picture, but still, the moment you see that picture, you can relate it to Mahatma Gandhi, right? So that's the kind of approach we need to take for the model development. Once you have such a model, the next stage is unify. That is, once you have such a model, you should apply to a wide variety of phenomena related to that brain structure. And you should be able to successfully explain all of them. So then you're able to unify all those phenomena under one model. And once you have succeeded that, it means you have a good model. It proves that you have a good model. And with that, you develop applications, both in neuroscience, like in clinical neuroscience, and also whatever is relevant to that phenomenon in engineering. And on a theoretical front, we would like to develop over the maybe next 10 years or so, a comprehensive oscillator theory of brain function, right? Because your deep networks are too artificial, they don't have oscillations, right? And the biological models are sometimes too detailed and too complicated. Between the two, we would like to develop models consisting of networks of nonlinear oscillators, right? And develop models of various aspects of brain function, like spatial navigation, attention, sensory processing, motor function, and so on and so forth. So I have my reference for this is this famous book by Georgi Bazaki called Rhythms of the Brain. I'm in touch with this. Is a wonderful scientist. He's at New York University in US. So I would like to realize whatever is described in this book in terms of experimental descriptions. I want to realize all that into theoretical form. And another idea is to test it on a robotic implementation, right? And so what we have these modules, different modules that we develop. We want to put all of them in a robotic implementation and try to develop a kind of a brain robot that works on a brain-inspired system. So long-term vision is to create a style of computational neuroscience modeling, which tries to integrate information from many different sources, and have developed these unified modeling models of different aspects of brain function, and develop disease models based on that, and then have dedicated centers which will look at single diseases or diseases of a very narrow class, right? And then develop startups based on that and develop clinical applications. This is a kind of long-term vision. And these are some of the alumni of my lab who did PhDs or masters, and the various universities are right now doing postdoc in various places like NIH, UCSD, and so on. Pragati has actually come back and joined IIT Kanpur as a faculty, and these are our collaborators, both national, international, and also industry collaborators. This is my lab. They look very happy, this picture. And Jai Hind. That's all I have. I mean, I would like to, spend some time in a discussion so that there can be some interaction. If you have any questions and if I can answer, I'll try to answer. So thank you very much.
Thank you so much, sir, for a really informative session. Participants, if you have any questions, please feel free to post it in the chat box, or you can ask yourself. We have provided you the mic access.
So I'm not able to open the chat box. Do we need some permission or something?
So for chat box, we'll read it for you, sir, if anybody posted in the chat box.
Fine. Okay, more questions?
Yes, sir. Hello, sir.
Yeah.
Good evening, sir.
Yeah, good evening. Hi.
So my question is that you have suggested there's some the excitement of neurons and how they are excite each other and how they are firing, sir.
Yeah.
So, sir, my question is that there is a reason behind for firing of neurons, like some metabolic changes takes place in one to both cells, like efficiency and their firing potential is to improved by using that phenomena, sir.
Sorry, I don't understand. So there is you can explain the firing activity, right, in terms of the ion channels and ion concentrations and all that. But what exactly is the question?
Hello.
Hello. Can you hear me?
Yes, sir. Yes, sir.
Yeah, you can explain the firing activity. There are lots of models for that. But what exactly is the question?
No, sir. My question is that we are using same concept for you have told that that is the these concepts are valid for the Parkinson's disease and all other Alzheimer's disease, sir. So can we apply this model for the ALS or MND kind of disease also, sir?
So we propose that the similar metabolic deficiency is the root cause for all neurodegenerative disorders. And
Yes,
so we have done modeling only for Parkinson's. But based on the experimental evidence that is already existing in literature, we just made a hypothesis and wrote a prospective paper on that. But whether it is actually true or not, we need to see. I mean, we haven't done the modeling for other diseases, right? And the real proof is if the metabolic deficiency is the reason, then if you can somehow replenish the energy necessary, then you should be able to arrest the progress of the disease. That wasn't demonstrated yet in experimentally. So we have to do that, and then only then that's a final confirmation that this theory is right or complete. So we haven't done that yet. I mean, nobody has done that yet.
Okay, sir. Thank you, sir.
But quite a likely hypothesis, but it has to be proven ultimately.
Okay, sir.
Okay.
Any other questions from the participants? So in the meantime, the participants are just coming up with the questions.
Okay.
So I'll just ask one small question. So one of your work is like modeling neural oscillations.
Yeah.
So there would be certain specific range of the rhythms which might be modeled, right? So what is the typical frequency range in that category, sir? Whether the high frequency oscillations are also taken into account, or like maybe you can just shed some light over it?
Yeah, so brain frequencies range anywhere from very low frequencies like 0.1 hertz to 1 or 200 hertz. It's very low compared to as an electrical engineer you're used to seeing gigahertz and all, but brain frequencies are very low. In our models, there are abstract models where omega is just a parameter, right? You can make it whatever you want, right? So, for example, for locomotion, it will be the order of a fraction of hertz, because these are this is a gate rhythms. But this is a modeling framework. You can make your omegas whatever you want. The real frequencies in the brain are in the ranges that I just talked about.
Okay, sir. Any other questions, participants? So I hope we don't have any other questions.
Okay.
So thank you so much for joining us today, sir. Indeed, it was really very informative, and we do hope that we'll have many opportunities in future to have an offline discussion, and we'll call upon you for further events. Sir. Thank you so much for
Thank you very
joining.
much, and good luck, everybody.
So there is one announcement for the Azadi Ka Amrit Mahotsav event. So we will be meeting again tomorrow at 5 o'clock by a session by Professor Amulya K. Panda. And for the BMAI participants, so we will not have any other session today. The last session would probably be done on Saturday morning 9 a.m. So thank you everyone for joining today. So let's meet tomorrow 5 o'clock. Till then, please take care and have a nice day.